I will present my work already introduced by the host. My name is Yasmin, and these are my colleagues of my laboratory. And I will present the PPIntegrator semantic integrative system for protein-protein interaction and application for host-pathogen datasets. Here is a brief summary of the content, beginning with the introduction, passing through the background concepts, motivation goals, follow with our proposed methodology to attain these goals, then proceeding to the results we achieved and the final remarks. Proteins are formed by a chain of amino acids synthesized from genes, and are essential molecules for all living organisms. Most proteins need to bind to another one to perform its function correctly, either in form of physical or functional associations. The protein interactions may occur among proteins of the same organism or among distinct ones, which is the case of the host pathogen subset of PPIs. PPIs are the abbreviation of protein-protein interactions and can be understood as a graph, where the nodes are the proteins and the lab detections or predictions form the edges, as illustrated in the figure uh, that is showed in the slide. And the main databases export their data following distinct patterns, and that's why semantic web standards plays an important role to standardize the data among databases. According to recent literature, there are few databases concerning host pathogen protein interactions data. Most of the PPI datasets do not export data following a pattern and did not adopt semantic web standards, and the few methodologies existing in this direction fail to represent all the data generated in PPI prediction experiments. Finally, there are few strategies towards to the combination of multiple PPI data sources with flexibility for the user's own data. These observed issues in the literature lead to the main goals of our work. The first goal is allowing the modeling of PPI data from distinct species. The second is developing a workflow to create new host pathogen candidate interactions and predict and perform in silico validation. And the last goal is building a tool that enables the standardization of the PPI data among the databases through triplification and enabling the integration to enhance the analysis. In order to accomplish the modeling of multiple species PPIs, we proposed an update to the existing ontology onto PPI. The left figure shows the main concepts of a previously published ontology for the PPI domain. It organizes the experiment of PPI prediction, the PPI datasets involved in the experiments, the interactions with the two participant proteins that are instances of a pair-component class, and each interaction may have values predicted from multiple evidence methods. The update concerns the pair-component class, which was incremented with the organism information, crossing with the respective taxonomic classification from UniProt. We also added support to the specification of literature articles supporting these interactions. To augment and generate new host pathogen PPIs, we used the well-known concept of guild by association from the validated pairs of proteins. We retrieved the respective patterns in the string database with a high confidence score of 900 and above, and then we proceed to generate pairwise combination with these direct neighbors. These interactions form the test dataset to be evaluated by a model trained with all known host pathogen PPIs. The newly predicted interactions from the test set were given as input to the PPI validation process to retrieve support in scientific literature. In order to combine multiple protein interaction datasets, we created the PPIntegrator that comprises two parts: data preparation and amplification data fusion. Starting with the data preparation, it receives data from four heterogeneous databases which are the hint, HPIDB, string, and our previously published workflow preprint. Hint contains validated interactions of human and other single organism PPIs. HPIGB is a validated interactions database, but for the host pathogen context, string is a great, great large database that comprises computationally inferred PPIs, and PredPrim is our previous published workflow to predict PPIs, and not only has the prediction of protein-protein interactions in a binary way, but also comprises the functional enrichment of these protein-protein interactions. This module integrates all available information of these databases and exports it in a standardized temporary format to serve as input to the next module. The typification data fusion module receives the interactions in the temporary format and distributes to the process following 5 steps. Typification means generating statements or triples in the format of phrases containing subject, predicate, and object. Like the two examples represent the interaction between RAB1A and NLEB1. The first step deals with provenance metadata concerning the interaction data generation and the PPI prediction experiment and its respective author. The second step reunites the functional annotations about the proteins for posterior integration, analysis, and inference. The third step generates triples combining the interaction pairs according to their respective score predicted by evidence methods of each data source. The fourth step executes the data fusion, remove duplicated entries of PPIs that may exist among the databases. Finally, the last step exports to a public and free database with the Lego graph that has an interface to run queries and view the results table. We evaluated the proposed methods using data from 4 pathogenic species of bacteria, which are Escherichia coli, Pseudomonas aeruginosa, Staphylococcus aureus, and Mycobacterium tuberculosis. These species have high clinical interest due to present mechanisms that promote their survival in stress conditions and enable their resistance to several drugs. The non-host pathogen PPI data for these species were retrieved for HPIB, and we can see comparing the number of PPIs at the table and the plot that there is still too few data concerning protein interactions among these species and human. The maximum number of proteins in the proteome is 2,889 proteins for Staphylococcus aureus, and besides there is 10,000 PPIs considered any host. When filtering for only the ones concerning human host, only 39 are kept. Not only there is a few number of PPIs considering host pathogen context, but also there are few high confidence partners for each bacteria in the string database. The string high confidence PPIs contains a score derived from text mining scientific papers, so we evaluated and compared this score with the finds for the same interactions with our method for in silico PPI curation. The E. coli bacteria have more evidence in literature than the other bacteria, since it's considered a model organism. The highest agreement occurred for the interactions with low text mining score given by both methods. And the causes for the disagreements is that the text mining score given by string is not updated along new articles are results in the PubMed central database. And another factor is that they consider as score the presence of the two protein names in the same sentence, not evaluating if the sentence indeed corresponds to an interaction context. While our in silico curation retrieves this relevant piece of information from the updated PMC databases in NSPI and contains strict rules to consider a sentence a positive PPI case. Here, we compared how much data was enriched in the interaction networks of the 4 species according to the original seeds, and E. coli and tuberculosis to obtain the highest editions of nodes and edges. Most of the new nodes comes from the host proteins. The newly predicted interactions and validated ones from HPIDB were given as input to the PPI integrator. After the integration, we performed a functional arrangement evaluation comparing whether the annotations of the new interactions are biologically coherent to the expected functions and mechanisms according to the bacterial infection in host cells. Here, I present the consensus of the most frequent annotations in the 4 species. Some of the most important annotations are the location, in which there is the integral component membrane, which traverses the phospholipid bilayer characteristic of bacteria. Furthermore, it's known that many proteins of bacteria need metal atoms acting as cofactors to perform interactions with the host proteins. When the bacteria infects the host cells, it generates inflammatory responses, recruiting some kinases proteins. They also need proteins with function of ATP and DNA binding to perform regulatory process. These findings corroborate the biological plausibility of the new interactions. The existing strategies to enrich host pathogen PPI data rely on complex mathematical models to form new connections based on the non-validated PPIs and do not provide a post-processing. And we use the high confidence direct neighbors to get the PPI candidates and add a robust predictor and in silico curation pipeline to add quality in the new predicted PPIs. Our proposal is advantageous but needs some known information to start the approach. The existing methods that form new connections for homologous organisms interactions could be combined in the future to attain the ones that lack of validated data. Concerning the semantic data integration, some methodologies use semantic data standards to model the protein interactions domain. Some provide an interface for query and analysis. However, only PPIntegrator proposes the adaptation of the most known PPI databases for typification and availability in the semantic web standards, also integrating functional annotations that already are in this data partner, such as genealogy, UniProt, and the database of pathways, metabolic pathways, KEDG. PPIntegrator also is flexible for customizing the user's input data and is able to model PPIs coming from single or multi-species. As the final remarks, we proposed the OntoPPI ontology update to allow the multi-organism PPI modeling. We developed a host pathogen prediction and curation workflow, as well a PPI semantic data typification integration workflow. Our analysis on the integrated data shows that the functional models are consistent with those expected according to the signaling pathways activated in the host pathogen interaction context for bacteria. Thank you. Any questions?
Thank you, Yasmmin. Any questions? Well, I have one. Yasmmin, I would like to ask you to which extent the triplification data processing for integration depends on expert knowledge, and to which extent this approach, your pipeline, can be extended or applied to other environments of host pathogen interactions.
Very good question. Firstly, yes, we intended to get the confirmation of known experts by the literature, and the typification data helps to give an explanation of the data of the interaction data. When we start to do this modeling, we are viabilizing other people from other domains of knowledge to understand this context. And by applying not only single and multi-organism PPI modeling, we also create space in the semantic web data format to run more specific and more enriched queries to analyze this data. Here, in this integration, we can have data from various databases, and we can get the expert of the validated interactions, the expertise of the validated interactions, together to confirm or infer more reliability on the predicted interactions from the predicted databases.
Okay. Okay.